Good morning, everybody. We're going to get started in 1 minute, and I'll go through the introductory slides. Okay, good morning, everybody. Welcome to the RCN webinar. This is our March webinar, and it is almost spring, so that's great for all of us. Our speakers today are listed here. So our first speaker will be Heather Bischel, who's an associate professor in civil and environmental engineering at UC Davis. She's going to talk about proteomics. And then our second speaker is Andrew Wang, who's a research assistant professor at Emory School of Public Health. He'll be talking about sampling design for wastewater surveillance. As some of you might have seen from our listserv, we are now putting all of our past and ongoing webinars on YouTube as a place to archive them permanently. This is the link here, and if you need the link again, you can reach out to us or do a search. We have a number of different activities going on, including a workshop and journal club, webinar nominations. If you'd like to see someone specific speak at the webinar, have a suggestion, as well as an email list. The links for those are going in the chat now by Tamina, who's the postdoc who helps us run the RCN, and she's instrumental to running the RCN. We're going to be having an upcoming workshop next week. You can scan the QR code to register, and the workshop is focused on vector-borne pathogens. And then we are having a coffee hour on Friday to talk about wastewater-based epidemiology, pathogen detection, and emerging methods and technologies. So please scan the QR code if you're interested in joining. And there's also a link in the chat. We also have a journal club coming up in a couple weeks where we'll be talking about a Candida auris study. Again, you can scan the link if you'd like to. I will not be there as I am an author on this paper, so you will be able to speak freely about it. And then we are having a wikithon. So the RCN is generating a wiki with publications and papers that are insightful and helpful on the topic of WBE. If you'd like to join the wikithon, please scan the QR code. This will be a session devoted to updating the wiki with papers, either your own or papers that you really like in this area. And then a final announcement is that in next month, we're going to have another Swiss-themed webinar. I'd love to feature another country. I think we would, but we have a lot of research going on in Switzerland. So we'll have a student from EPFL talk about a survey that she put out about wastewater and perception of using it for monitoring. And then we'll hear from someone who works in public health in Switzerland about data aggregation. So with that, I will stop sharing my screen, and then I'm going to have Heather share her screen and we can get started. Sorry, that introduction took a little bit longer. And I already introduced Heather. I'll just let her take it away here.
Hi, good morning, everyone. You hear me okay? Yes. All right. Well, thanks so much for the invitation to talk today. I'll be talking about a proteomics investigation that we've been doing of viruses in wastewater and focusing on SARS-CoV-2 peptides and especially mass spectral signal normalization factors. And this work was done by my postdoc, Wen-Ting Lee, and in collaboration with the Lawrence Livermore National Lab, Carolyn Fisher, and then the UC Davis Proteomics Core Facility with Gabriela Gregorian. And I want to take a second to thank you all for the, coordination network and all of the amazing collaboration that you've inspired amongst many. My own kind of applied work in wastewater epidemiology started with a hyper-local and very intensive public health investigation with Healthy Davis together. My lab still runs SARS-CoV-2 analysis for our campus wastewater treatment plant. So UC Davis has its own wastewater treatment plant, which we've used quite a bit to develop new methods and work on different aspects. And we expanded out Healthy Davis together into the Central Valley of California to focus on agricultural, rural areas and disadvantaged communities. And we're really fortunate to be a small part of the wastewater scan network and supporting a community of practice in that region. But the work that I'm talking about today is a project we've had going on over the past 3 years. You've had Rose Cantor and Minji Zhang talk about some of their work on sequencing. It's called Preview: Pandemic Readiness and Equity Through Virus Epidemiology of Wastewater, and it's UC Berkeley, UC Davis, Lawrence Livermore National Lab, and UC San Francisco. Part of that project, a big part of that, is to benchmark new methods for novel viruses. Of course, I'm not talking about novel viruses with SARS-CoV-2, but we wanted to focus on using SARS-CoV-2 as a way to develop proteomics. And we all know PCR is gold standard for pathogen quantification in wastewater. And we've also seen that targeted mass spectrometry can be very reliable to monitor chemical exposures and substance use patterns. Highly targeted, specific, sensitive analysis for strong quantification, established and widely available, highly sensitive. Having analysis one at a time, multiplexing can be difficult. We see that being done. We know that we need to know information about the sequence or the structure in advance for targeted analysis. And we can see some decreased sensitivity with mutations and chemical transformations. So we wanted to look at how wastewater proteomics can be a complementary tool to the high resolution trend analysis that PCR and mass spec targeted analysis can provide. Proteomics is used in a wide range of applications to assess the expression of genes of interest and the proteins associated with those genes, and could be used in assessing biomarkers of chronic disease and community health, like inflammation-related proteins. So our research objectives for this aspect of the preview project were to benchmark detection of SARS-CoV-2 derived proteins, focusing on the spike protein as well as the nucleocapsid protein, to enable extension of wastewater proteomics to other biomarkers, thinking more broadly than SARS-CoV-2. And we wanted to work to distinguish variants of SARS-CoV-2 based on the amino acid sequence differences in the spike proteins detected in wastewater. And then we looked into improving the quantification potential of mass spectrometry for proteomics by evaluating different signal normalization factors. So our overall approach, I won't talk much about kind of the method development aspect. We focused on increasing peptide yield, removing interferences from wastewater. We know it's a complex mixture with matrix effects from large macromolecules and other large and small molecules. There's, of course, always concern about signal decay with proteases or low abundance targets of viruses relative to other molecules, and of course signal variability in the sampling as well. So our method development was based on some literature studies that were available for wastewater, and then a lot of expertise from the proteomics core facility at UC Davis. And once we settled on the method that we wanted to use, we conducted a series of validation experiments. We had 21 recombinant protein standards that we used, both N and S proteins, as well as 3 inactivated whole virus standards. And then we had a short study, a 2-week daily study where we applied the method that we validated to untreated wastewater, trying to quantify the relative abundance of N and S peptide levels and to benchmark the results against our ddPCR analysis for N over PMMOV results at the UC Davis wastewater treatment plant. So the method that we settled on for this project, and as I said, we learned a lot of lessons from a few wastewater proteomics studies that have been published in the last several years. We sampled influent from our wastewater treatment plant about 50 milliliters, and we decided to separate the influent into two fractions using a centrifugal filtration device, a retentate fraction for molecules larger than 10,000 Daltons, and then a filtrate fraction that was for smaller than 10,000 Daltons. And the idea there was that we thought the filtrate fraction, we had either pre-digested peptides or smaller molecules here that we could process and clean up through solid phase extraction without additional digestion steps. And the retentate fraction, we passed through an S-trap proteomics sample preparation protocol for digestion and generation of peptides. So there was fewer processing steps here that we wanted to look at the potential for identification and quantification in those two different fractions. We used peptide retention time standards and protein digestion mixtures for mass calibration and for validation of the instrument, and working really closely with the Proteomics core facility and also at Lawrence Livermore, testing various instruments, but landing on the Thermodynamics 3000. So data analysis that Wenting worked on kind of had two different components using FragPipe. One is looking at data-dependent acquisition. And the idea with data-dependent acquisition is kind of a more standard or traditional method for proteomics, where you've got to scan across different mass ion events, and the highest abundance ions are selected and isolated and then fragmented into smaller molecules so that you have a good, what you might call a clean MS2 spectra that eliminates issues of co-eluting ions. So this method helps construct a MS-MS spectral library. And then there's data independent acquisition, where it's a scan that takes all of the ions in across various MS2 window sizes and isolates these fractions of the spectra, but it fragments all ions that are within that window. And so you, instead of picking just the most abundant peaks here, you're looking at all of the peaks present. So the nice thing about this is you do have a messier MS2, but you're capturing all of the ions. So we decided to, for all of our samples, run both a data dependent acquisition and a data independent acquisition. And then we used Diane for quantification and analysis. So that DDA helps us support the spectral library development and peptide identification, whereas the DIA is more reproducible in terms of quantification, because you can have differences in the relative abundance of different peptide peak areas. And then it facilitates extension to new targets as well. So it's a non-targeted data acquisition method where you can look back at these large data sets that you collect and start to assess other peptides that are present other than the ones that we're looking for. So kind of as a big picture overview of what the sample results looked like. So that retentate fraction that we were looking at, we detected almost 14,000 peptides in the sample. The gray area, and the circle represents the full sample, and the gray area is unclassified peptides. You can see we had some archaea, some bacterial peptides, eukaryota, and a few, 117 peptides that were associated with viruses. So, and this was using a Swiss Prot database that has over 500,000 sequences. And then we augmented that database with sequences from 32 SARS-CoV-2 viruses so that we could assess SARS-CoV-2 in particular. In comparison, the filtrate fraction, which we expected to have fewer peptides associated with it, had about nearly 4,000 peptides. So we had a lower peptide yield in the filtrate, but a higher proportion of viral peptides compared to the retentate. So kind of a similar number of viral peptides in that filtrate fraction. But we wanted to focus on this retentate fraction. So these were peptides that we assessed, and then those corresponded to about 4,000 proteins and 1,300 proteins in the filtrate. So relative to one of those past studies, we had increased the yield of the peptides. They had found about 3,300 peptides in wastewater and a combination of a soluble fraction and a pelletized fraction. So, we had some improvements on the method. There's still a lot more to be done in terms of mass spec methods to improve sample processing pipelines and more. So in terms of the retentate fraction, 77% of the peptides were identified in that fraction, including 8 variant-specific peptides. And I'll talk more about our validation to look at the feasibility of mass spec for variant differentiation. We use that data to assess candidates for normalization factors. So we had a couple hundred human proteins detected. We looked for ones that were high abundance and high frequency detection. There were 3 that we focused on, glucoamylase and a couple of alpha amylases. And I'll come back to that as well. Those were present in all of our samples with 50 to 100, more than 100 peptides associated with each of those proteins. So our variant differentiation validation, we use SARS-CoV-2 recombinant proteins from historic variants that weren't in circulation. We detected from spike-in studies 114 peptides for those S proteins, had similar coverage across the spikes with conservation mostly across those different variants. And as I mentioned before, 8 variant-specific peptides we were able to detect here as well with a couple unique peptides per protein standard. So this was a demonstration to us that, variant differentiation is feasible with the mass spec processing pipeline that we were working with when you have spiked in samples. One of the key challenges we wanted to try to address was building towards a semi-quantitative proteomics pipeline. So with S proteins, again, we spiked in recombinant protein standards, detected peptides associated with that. We worked with a heavy arginine internal standard for our normalization of those peak areas to calculate protein abundance. And we were able to generate linear calibration curves for 36 peptides associated with those 5 spike proteins with a dynamic range from 40 to 1,200 or 2,000 micrograms per liter. We also looked at that kind of semi-quantitative potential for N proteins. So there was actually really high abundance of S proteins in the wastewater from the method that we were looking at. So we were in trying to calculate a limit of detection and a limit of quantification. We focused on the N proteins and did a dose-in study over a range of concentrations, looking at the protein abundance compared to the protein abundance in the sample compared to the spiked in mass. And then we calculated what theoretically those masses might correspond to for some different variants in terms of genome copies per liter from whole virus SARS-CoV-2 standards. And certainly the limit of detection from our theoretical calculation compared to the genome copies is pretty high. So several orders of magnitude higher than what you might get from a targeted PCR type analysis. Certainly there's more work to be done in terms of increasing the sensitivity, but we were seeing good detection of both N proteins and S proteins in the wastewater background. So we applied the method then to compare the relative abundance of N proteins and S proteins in the wastewater. What you can see here in the normalized internal standard normalized peak area, the S peptides were consistently more abundant than the N peptides in our kind of short daily analysis. We had 79 peptides that corresponded to the XBB 1.5 variant at the time of the study, and 1 high abundance unique peptide shown in black here that was unique to the XBB 1.5 variant from our searches. Problem is that at the time, XBB wasn't the dominant circulating variant. It was JN.1. So this was the dominant circulating variant at the time. We had 1 peptide detected associated with JN.1 that was an N peptide, and it wasn't unique to JN.1. But what we know is that that same time, in September of 2023, CDC had recommended monovalent XBB 1.5 COVID-19 vaccines for all individuals age 6 months and older. So there was a vaccination push going on for this XBB 1.5 variant. And of course, these are primarily mRNA vaccines that cause the expression of spike protein of the XBB 1.5 variant. There's uncertainty as to the duration of the presence of the spike protein in humans and its ability to be excreted, but we postulated that these S proteins and some of them could be associated from vaccine-derived sources. In terms of quantification, though, we wanted to then focus on the N proteins because of that confounding factor with potential vaccination-derived proteins, even though that also represents an opportunity. So coming back to those 3 human proteins in wastewater that we had detected at high abundance, high confidence in their identity, and very frequently in the wastewater from UC Davis. Here you can see in the same time frame of analysis, the alpha amylase fluctuation through time. We had at least 47 peptides in each of the samples detected. Alpha amylase 1B here, 12 peptides per sample, and this maltase glucoamylase as well. So these were our 3 candidates, and I'm showing them side by side here with our ddPCR measurements for pepper mild model virus in the samples, which we have a series nanotrap influent extraction protocol to measure that. So same sample, different processing method. We weren't able to detect very many peptides associated with PMMOV. It's hard to explain in terms of ion suppression, matrix effects, extraction, database availability, exactly why, but we weren't able to use this as a normalization standard and focused on these more high abundant, high frequency peptides and proteins. So then looking at the opportunity for comparing non-normalized and normalized samples, this is a little tricky to explain this particular analysis, but we spiked SARS-CoV-2 whole virus into 2 different wastewater samples, and we evaluated the effect of the normalization on correlations between the differences between spiked and unspiked samples for each peptide that was detected for the whole virus spike-in samples. So the abundance of each of the 29 SARS-CoV-2 peptides that we detected, removing background, and doing the same thing for virus spike peptides, but normalized by one of the normalization factor proteins that we were looking at. So here you can see we'd expect a one-to-one relationship between the first wastewater sample and what was spiked in for each of the peptides that are shown as dots in the wastewater sample 2. Expecting that correlation, not seeing it without normalization. And when we incorporated in the normalization with either of the 3, any of the 3 proteins, we had a very strong improvement in our correlations between those 2 sample results for the spike-in. So then we wanted to apply those results again in that same short longitudinal study. We measured SARS-CoV-2 N gene RNA concentrations normalized by PMMOV, measured by ddPCR. And then we assessed the SARS-CoV-2 nucleocapsid protein normalized by alpha amylase 1B, as well as the other targets. We had kind of similar results between each of the targets. This seemed to be our strongest candidate, where we had a moderate to strong correlation between the results of our semi-quantitative proteomics pipeline with SARS-CoV-2 nucleocapsid compared to the RNA concentrations of the N gene over that sampling period. So kind of wrapping up here, we did see this increased peptide yield in the retentate compared to some past studies. There's, of course, more room for improvements. That non-targeted data acquisition facilitated our testing and selection of amylases as signal normalization factors. The relative quantification of N and S proteins was consistent with our expectations of higher S peptides relative to N, given viral properties, and did have that ubiquitous presence of the S protein, and some question about the source of the XBB 1.5 specific peptide that we detected. And then we applied those methods, and we successfully benchmarked the N protein quantification against the N gene measurements. And we think there's opportunities, to extend these methods to new targets, and of course look across a broader range to assess the repeatability of these normalization factors in other locations. So thank you all again, especially Wen-Ting Lee, who's led this work. The project's funded by the UC Office of the President, and I want to thank Cara Nelson, who's the PI for the project, and Rose Cantor, who's a project manager, as well as our partners at Lawrence Livermore and the Proteomics Core Facility, and some of my lab team members who supported some of the work as well.
Thank you, Heather, for that great talk. And I think this is the first talk we've had on proteomics. For the audience, please put any questions you have in the Q&A. So I'll start off with a question, and I see there's one already in the Q&A. Please add more if you have any for Heather. What is your sense of the persistence of these peptides in wastewater? And are you planning to do any work to better understand how they persist, especially relative to some of the RNA targets, because those have been surprisingly persistent in wastewater? What is your sense about protein persistence in wastewater?
Yeah, my sense is that we expect some good persistence, right? We are doing some studies. So also as part of the preview project, I have a PhD student, Jordan Beck, who's working on looking at proteases in wastewater, so kind of wastewater-derived enzymes, and how degradation of viral proteins on the surfaces can, cleave some of the peptides present, and looking for signals of those in the wastewater itself. But even with degradation, you do have many peptides that you're working with a given, large biomacromolecule. And one of the previous studies had looked at PPU1AB. I may mispronounce the name of the target, right? But it was this relatively large protein that, some of the external structures maybe are degraded, but, you could still be detecting some of the internal components. And so with proteomics, and you're kind of deconvoluting these signals from many peptides to be able to tell you something about the overall protein abundance. But we did, at least in the context of our study, have, high numbers of peptides associated with those human proteins and consistent detection there. But I think, further validation elsewhere in other contexts is certainly necessary to look at the consistency.
Yeah, thank you for taking on this really complicated project. It seems like one of the dirtiest systems possible to work in for proteomics. We have a couple more questions, and we won't have time to cover them all, but I'll ask one from Marlene Wolf. She asked if you could talk a little bit more about the mRNA vaccines, and if they're increasing use, assuming in the future that we use them more for different viral vaccines or other vaccines in general. Is this a problem for using proteomics in the future? Like, does it represent sort of not like something that could prevent this from ever being used into the far future once it's all worked out?
Yeah, thanks for that question. And I actually think it's almost more of an opportunity, and I don't want to overstep from our results necessarily, right? But when we were looking at this, you see spike proteins that do seem to be associated with that from vaccines. And when we're talking about ddPCR and nucleic acid-based signals that are very reliable, we're not trying to compete against methods that work really well, right? What are some complementary uses? And in this case, right, you wouldn't be able to detect the mRNA signatures from the vaccine likely that are, degraded more rapidly of naked mRNA compared to some of the protein signatures, and then the presence of the spike proteins from potentially from direct vaccine-derived sources could start to look at, vaccination through, successful vaccination strategies in different communities, and provide kind of a complementary tool in that sense. Of course, there would need to be much more validation in terms of are the levels that how consistent is that detection from one vaccine, formula to the next? How does that relate to vaccination implementation? I think what we're looking for are some of the opportunities, but not necessarily to replicate what PCR does really well.
Yeah, that makes sense. So I think we have time for one more question, which is, can you speak a little bit more about what you're thinking about why PMMOV peptides weren't detected? And I know you alluded to the fact that you don't know, but given that those peptides are missing, you know, what other viral peptides are missing, and why are some missing and some not missing?
Yeah, I think, because we had such strong detection, and right, we were looking for what are the signals that are present at those highest, levels, and the extent to which human proteins are incorporated into databases, right? Sort of the relative presence of certain families of viruses into the protein databases compared to others, right? We were working with what was in Swiss Prot at the time, so I think there could be some aspect of it that's related to the databases that we're using to pull out what peptides are associated there, in terms of mass spec ionization efficiency, or how compact or small different peptides are and their incorporation in the solids versus in the liquids. There's a lot of questions around that. So I don't have a good answer necessarily for that. And yeah, we did expect to potentially see more. PMMOV is so commonly used, as are many other proteins, right? So there's more work to be done there, but we did have that kind of high confidence on the ones that we detected that did seem to be good candidates.
Thank you so much, Heather. We're going to wrap up and move on. I'll pass it to Ralph, but thanks again for working on this really complicated problem. So keep up the good work and don't stop.
All right. Great work, Heather. Yeah, so compliments to you and your group for doing this difficult work. Very exciting. So we have set the bar pretty high, Andrew. So you're going to be next, Andrew Wang, Research Assistant Professor at the Hubert Department of Global Health at the Rollins School of Public Health, Emory University. And he's going to talk to us regarding strategic sampling design for wastewater surveillance. Take it away, Andrew.
All right, thank you. Let me share my screen. Can you see my screen?
Yes.
Okay, great. Thank you, Ralph, for the introduction and then the opportunity to talk, present this work in this webinar. My talk today is about the strategic sampling design for wastewater surveillance. Okay. So I'm going to skip the basic background information about wastewater surveillance, as the audience for this group are very familiar with this topic. Let's directly cut to the point. So what should we consider when it comes to wastewater surveillance sampling design? So we have a list of key factors to consider. First, you want to think about what's the goal of your surveillance, whether it's for early warning, or it's a temporal trend monitoring, or spatial hotspot detection, or for targeted population monitoring. And what's the pathogen and the biomarkers you're doing for the surveillance? And then what's their prevalence? What's the shedding information? How is their persistent in the wastewater? And then what's their sampling methods, whether it's a grab sample versus a composite sample, and then the sampling site selection, sampling downstream compared to sampling upstream, and also sampling frequency, whether it's like very frequent, like a daily, or less frequent as weekly. And last, we also need to consider the logistics and implementation of your wastewater surveillance. Consider is safety, flexibility, and sustainability. So in this talk, I'm going to focus mainly on the sampling site selection part. So first, let's check what's AI's recommendation, as this is more and more popular as one of our first solution. And here's the answer from ChatGPT. It outlines 4 steps, including define surveillance goals, identify key locations, consider practical logistical factors, and use multi-tiers approach. It's a pretty good answer, but I think we can definitely do better than this. So here's our proposed strategy. Number 1, design wastewater surveillance as a system. So why do we need to design wastewater surveillance as a system? I consider this as a scenario of the plant and the elephant. So each wastewater surveillance sampling site can be a plant man. They observe the elephant from different angles and could have different views. So the this plot, this figure on the left is modified from one of the figures from sketch planations. And then I modified this a little bit to show those three different blind men kind of touching the elephant's different part of their body, and then thinking about this as a disease trend. Some people will think about the surging as they're touching maybe the nose part, and then when they touch the belly part, they think it's flat, and then when they the another one touching the tail, think it's decreasing. So they cannot convince each other, and then they cannot agree with each other. But when they are collaborate together, they should be able to identify the elephant together. Okay. Next, I'm going to talk about some cases to illustrate this idea. First, I'm going to talk about our multi-level COVID-19 wastewater surveillance in Atlanta. So at the city level, 362 grab samples were collected from 9 effluent line sites entering 3 wastewater treatment facilities between March 2021 and April 2022. And then by the time we have the sewer network data from the city and then the geocoded COVID-19 cases data from the Georgia Department of Public Health. And then the map on the left shows the sewer shed of those 9 effluent line sites. We were able to examine the correlations between the concentration of SARS-CoV-2 RNA in wastewater and the number of daily reported COVID-19 cases, both at county level and the sewer shed level. The figure on the left, the red curve shows the county level reported cases in Fulton County, and then the yellow line shows the reported cases at the sewer shed level. And then those points, the purple points, or our measured concentration of SARS-CoV-2 in wastewater, and then we smooth it for each individual sampling sites. So what we found is the correlation actually varied. As you can see, the Spearman coefficients on the top right corner, they actually are different between different sites. But when the temporal trend of disease in the city become clear when examining the results from all the sites together. And the community level, we collected 506 more swab samples from 56 community manholes around same time period. And there are 3 phases of the sampling design. First, we started with some convenience sampling. And then once we get the sewer network data from the city, we move to a more like a targeted catchment size sampling, trying to understand the big sewer shed versus small sewer shed sampling. And then at last, we kind of settled down with a nested sampling design, which I'm going to talk more details in the later slides. So what we found is in phase 1, the detection rate of SARS-CoV-2 of those community manhole sites varied even between manholes in close proximity. And in phase 2 and 3, some sites consistently detect SARS-CoV-2 RNA, while others only detect positive when there's a surge in the reported cases. So this is a kind of the idea: you have different sites, but you have a variations of their sensitivity, and then they may observe different signals. However, when the percent of the positives across that together also show the matching trend, which is this plots in the middle with the reported cases, which is the plots on the bottom. So to integrate the city level and community level wastewater surveillance, our strategy is for phase 3, this is nested sampling. We set up a nested sampling design by sampling one downstream influence line site along with multiple upstream community manholes with independent catchment areas. The map down here shows the whole, the entire area is the sewer shed of one influence line site, Philip Lee. And then we also have 5 upstream community manholes catching a sub area within this sewer shed. So such a design enables us to track the temporal trend and also identify the spatial hotspot at the same time. So this is validated by observing the catchment area of the sites with a high SARS-CoV-2 detection rate in the wastewater in the subplot A, which is this pink area, and then this blue area, overlapped with area with high numbers of reported cases, which is in the subplot C. And then this is the spatial distribution, or you can see a heat map of the geocoded cases. And then there's another small case, and this is the institution level wastewater surveillance on Emory campus. So between July 2020 and March 2021, the wastewater surveillance samples were collected from 21 student resident homes on Emory campus. And then the map on the left shows our the distribution of those sampling sites. During the spring 2021 semester, large proportion of the wastewater samples across the campus were positive 1 to 2 weeks before the surge of the cases detected by the clinical diagnostic testing. So having multiple sampling sites in the same wastewater surveillance system enables the confirmation of early warning without you have to go back and collect additional sample for confirmation. All right, next, let's move to the strategy number 2, which is a design wastewater surveillance as a dynamic system. So I'm going to use the case of typhoid wastewater surveillance in Calcutta to illustrate this. In 2018, we collaborated with National Institute of Cholera and Enteric Disease in Calcutta, India, to design and conduct wastewater surveillance for typhoid in 2 wards in Calcutta. The 2 wards has about 160,000 people and about 30,000 toilets. And then the shared toilets are common in those 2 wards. The sanitation system is the underground sewage system, and most of the sewage pass through the pumping station on the north boundary of those 2 wards. However, when we were trying to design the wastewater surveillance, we faced a lot of real-world challenges, such as there's a limited sewer network data, and they are not digitalized. The plots on the left shows the map I got from our partner, showing what the sewer network looks like. And then the sewer networks are underground and are not observable from the surface. So what we do is for this sampling design, we use a simulation study to answer questions such as whether you should sample at the pumping station or toilets. If you're sampling on the upstream toilets, which ones you should sample? And then can the selection of the sampling site be improved during the wastewater surveillance process? On the left is the illustration of our simulated sewer networks. And then I'm going to quickly go through those slides. So in this simulation, we build a mathematical model to simulate how the shaders were generated, how they were assigned to toilets, and then what their shedding kinetics, and then for how long, and then for typhoid, there's an intermediate shedding, like whether they not they're shedding on that day, and what's the number of similar typhi they shed in their stool samples. And then we also simulate the pathogen fate once the pathogen go into the sewer system, how they flow to downstream, and what's the decay and the loss during this process. And last, we also simulate multiple type of the sampling strategies in this, and then compare to answer those questions we mentioned before. So using this information from the simulation study, we made a decision tree to determine whether we should sample upstream and the toilets, or the primary sampling units, which is a cluster of toilets, or downstream and the pumping station. This question answer, the first question, whether we should do downstream or upstream, but this does not really answer which toilet we should sample and how to improve the sampling design over time. So our proposed strategy is adaptive sampling. So basically, this has 4 steps. First, you have to initialize your sampling by dividing the geographic area into certain number of cells, and then randomly select like a primary sampling unit from each cells, which is those small rectangles in those in the map in the plots. And then you collected several rounds of samples and each sampling site. You apply a jackknife approach to the results collected by removing one sampling site at a time and assessing the corresponding information loss. So the basic idea is the more important a sampling site is, if you remove this and then if you lost a lot of information, then this sampling site is more important compared to the others. And then this loss function can be totally customized by the users. And then the sampling site with or the PSU with the smallest information loss is going to be relocated to another site, either by expert opinion or randomization. And then let's take a look at the results. So we have the base map of the sewer networks, but which is unobserved in Calcutta. So this is purely from simulation. And then the size of those red points indicate the risk, disease risk in the area. And then the counter plot shows the spatial distribution of the sampling site in 1,000 simulations. So during update 1, when you initialize, they're more like uniformly distributed based on this initialization steps. And then as the update steps goes on, what we can see is the sampling site were relocated to those high risk areas. And then the detection sensitivity of wastewater surveillance increase. Okay. And next, let's move back to Atlanta because it's a different scenario. Unlike Calcutta, we do have the sewer network data in Atlanta. For COVID-19 wastewater surveillance in Atlanta, we combined the nested sampling and adaptive sampling together by relocating those community manhole sites within a nested structure, which is the example was showing on the left. And then let's see. In this simulation, we create a hypothetical high-risk area and the left top corners on the sewer network. And then we initialize this system with 1 downstream set here, and then 5 community manhole sites. And then as the update goes on, you can see the estimated risk, which represented by the size of those nodes in the network on the left, top left corner increased with those updates. So to summarize this part, designing wastewater surveillance as a dynamic system is critical for developing a sensitive and actionable wastewater surveillance. And the nested sampling strategy with an adaptive sampling process enables the identification and the tracing disease hotspots in one area. So, at the end of this, I want to take the time also to show you guys some other efforts we are currently doing to support the wastewater-based epidemiology. Just quickly 2 or 3 slides. So previously we had been focusing on the sampling part, which is the yellow part, thinking about where to sample, what's the sample frequency, what's the method you should use, using a lot of simulation study. But moving on, right now we're exploring more about the population side, including the shedding kinetics and then the impact of the population mobility. So I'm going to show you 2 quick project we are doing. This one is the first one is a shedding hub. So we created an open science project called Shedding Hub to aggregate, standardize, validate, and doing analysis for shedding data from published studies. It includes a public data repository, modeling tutorials, and the data dashboards. The screenshot on the left is our website, and then on the right is the example database after the standardization. So this work is a collaboration with Dr. Till Hoffman at Harvard, and we welcome contribution and collaboration. So please feel free to contact us if you are interested. And then we are also working on integrating large-scale agent-based models, including pattern of life, which is population mobility and their social contact, infectious disease dynamics, fecal shedding kinetics, and wastewater surveillance. So this work is led by Dr. Andreas Zufel and me and Emory. We plan to use the model to explore how wastewater surveillance can guide the public health policies under different scenarios. So on the left, this is a plot from Andreas' paper. And then you can see examples of how we simulate population mobility in French Quarter, New Orleans. And then there's social network, which is the top right corner of this plot. And then the disease dynamics, which is the plot in the middle, so showing the number of infections. So we're planning to add this one, integrate with the wastewater model and the shedding model we're creating, and then create a big playground for all of us. Okay, and then that will be the end. I want to acknowledge the support we had from our founders, Bill and Melinda Gates Foundation, NIH, and the partners, and also all the hard works by the amazing Emory team, led by Dr. Christine Mo. Thank you, and I'm happy to take any questions.
Thank you so much, Andrew. Great work in very interesting location. So I'm particularly intrigued about the work in the developing world, which is always challenging. Maybe before we get into the questions, you mentioned ChatGPT, and it's probably a good idea for all of us to always run that first and see what the computer says we should be doing. But the work is really incomplete if we don't, in the end, compare what ChatGPT fed us to where we ended up, right? So you did an analysis and felt that, yeah, you can do better. But I think we are then obligated to actually demonstrate that whatever we produce is more valuable, right? That's probably an exercise that we all should do at home for any future work to justify our existence. I leave that as a comment. And maybe let's move on. There was 2 questions regarding sample frequency. One, I think you answered with one of your slides. The question was, is it worthwhile to do less frequent than weekly? And so maybe you want to answer to that?
It depends. I think, you know, for wastewater surveillance, especially like in low-income countries, there's a lot of on-site sanitations. And then when you're doing sampling, for example, like those pit latrines or septic tanks, if you collect like daily samples, there's a lot of huge autocorrelation. Means, you know, one of the shedders may shedding for maybe 2 weeks, and then the signal in their septic tanks may last for months. So in that case, I will probably thinking about increase the number of sampling site and then decrease the sampling frequency. So this is totally strategic, depends on your surveillance goals. If you want to do early warning, for example, we are focusing on this term like early, then more frequent samples should be, you know, should be kind of needed. Then once you get the signal, you can immediately turn into actions. But if you just want to, because there's like in those low-income countries, the clinical surveillance is not very well sustained. There's nothing else you can compare. Then in that case, I think to provide at least some information to understand what's going on in the community, I think less frequent sample but covering larger population or larger area probably will be the way to do it.
Yeah, and I guess it also depends on the agent you're looking at. If it's a long lag in infections, you know, if it's slow incubation period, then maybe longer periods work. Also, if you have silent epidemics, right? So if you have an epidemic that's happening and the healthcare system isn't really prepared or able to monitor those, you know, doing that monthly is better than having no data on whatever might be completely undetected by other methods. Cool. And another question was related to the characteristics of your study site, particularly, I mean, if you look at the developing world, there's a huge gradient from, socioeconomically, from populations to, maybe the really disadvantaged and poor. So the question here is, if you have information on population and sewer system characteristics, would it be prudent to study those and integrate it into the selection of locations?
Yes, of course. So I'm going back to one of my slides. So, and during the simulation, we assume, if we do not know anything, then we use totally randomizations. However, if you have a priority for certain, areas you want to, place or prioritize to place your sampling site, you can also relocate those based on which I make like expert opinion or some other things. And also in this case, the loss function, which is here, this I, is totally customizable. If you want to put more weight or more priority on certain variables or for those areas, you can totally do that. So it's totally customizable. We're in this first round, because nobody has done this before, we just totally use very general and simple scenarios, but all those things should be able to be done based on the local scenarios you want to do.
Yeah, thank you. There was one more question regarding whether it makes sense to reduce the sampling frequency based on and off season of certain pathogens. I think that's almost self-explanatory. If you know that a vector isn't there, for example, then it wouldn't make much sense to monitor, and you obviously step up your efforts when you know that the flu season or whatever starts. But maybe let's switch and examine your location. So Kolkata, one of the large multi-million city mega cities. What did you learn in 2 minutes from working in that environment, which is so much more challenging, including all the logistical shipping samples and so forth? What is your kind of experience and word of advice as we venture on with the technique?
I think maybe my, Dr. Christine Mo, who I think she is in the audience, probably can answer this better. But I do feel in those settings, there's a lot of challenges when to set up the surveillance and get it running. Because a lot of times you don't really have the equipment, and then once you get, maybe there's some shipping issues, then you cannot sustain the enough like the agents for your sampling. And then you also need to consider the logistics in those cases, because even though I show this map in this like a very small area, but when you think about this huge city, the traffic time come from one side to another, so you also need to think about the manpower, the schedule of the sampling to sustain this. All of those need to be carefully planned, because from my end, I do a lot of simulations. I don't really consider those, but in the real scenarios, you have to have this discussion with your field teams back and forth to really settle down to a final sampling plan. Because I can always propose what I think would be the best, but sometimes in the reality, it just cannot be done in that way. So you need to compromise and then keep the communication. But I do feel the communication part with your partners, local partners, is critical, and then this should be done all the time.
Yeah, I wouldn't want to start a study without talking to your team. And I'm just thinking of a world map. If we had, science contacts for different regions of the world where such work has been connected, it would be hugely helpful for everyone to, really get the best out of their research dollars by talking to people who understand what's actually on the ground, how quickly, stuff can be moved and whether it can be shipped and all of that. And of course, when you're local, you know all that. If you're not, you make assumptions that oftentimes, are horribly wrong. Wow. So there's a lot of, these were 2 really strong talks. So I, applause here to the presenters as well as the series here, which seems to be getting better all the time. Before we sign off, I just want to mention again that next week on March 13, we have the RCN vector-borne disease. If you have it in you, it's going to be a 2-hour exercise. And so we hope you sign on. There were QR codes present. I'd like to, thank everyone who dialed in. And if Ali isn't here, so I don't know, is this Tamina, do you want to have a final word, or are we good to sign off?
Yeah, we can.
Yeah. So thanks again to everyone presenting for joining, and I'll see you next week. Bye.
Thank you.
All right, thank you. Bye.